Well, thank you everyone for coming out to what I've been up to for the past couple of years. My master's thesis research focused on investigating the microbial associates of algal symbiont-bearing foraminifera from the Florida reefs. And so today I'm going to tell you a little bit about some background information that will help you understand the conclusions that I've drawn from the data, introduction to the hypotheses I've made about the foraminifera, introduction to our methods, results, discussion about what we found, conclusions, and then directions for possible future research. So first and foremost, you need to know what a microbiome is. And as genomic research becomes more affordable and consequently more widespread, we're now understanding that an organism is more than meets the eye, because it's not just the organism itself. It's host to many different life forms, including bacteria, archaea, fungi, and in some cases, like my study species, symbiotic algae. And so the term for the collection of the host and those associates is termed the holobiont. And the microbiome is a subset of that holobiont in that it is the collection of microbial associates of that host. And it's unique to each organism, so it's kind of like a little fingerprint, if you will. And just as we're understanding the main factors that influence human microbiome, such as where you're born, how you live your life, your diet, we are also understanding that marine organisms have factors that are influencing and shaping the microbial community that lives inside of them. And some of these have been recognized as host species in corals and sponges, as well as disease and bleaching can shape the microbial community. So why do we care? Why do we care about these little itty-bitty creatures living inside of us? As you can see from this image, just your gut bacteria alone can help perform many useful tasks for you, including synthesis of vitamins, immune support, all those good things. And so the functions that your body is performing is actually a mixture of your body itself and your microbial associates. So it's really important, and they're necessary to perform the vital functions of life. Some of the roles that are hypothesized in corals and sponges, and I bring up corals and sponges the most because they are the most widely researched marine organisms with respect to microbiomes. So some of those functions include protection from disease, as well as synthesis of nutrients and biogeochemical cycling. And as well, these relationships can be evolutionarily advantageous because, as I said, they perform these vital functions, and as well, they provide the host with a mechanism by which it can actually adapt and evolve to changing conditions much faster than a higher order animal usually can, because its microbial associates can change much quicker because they have shorter generation times. So foraminifera. So what are these little guys? So these are unicellular amoeboid protists. And they make a little shell, and in the words of Ben Ross, as I love his description, they're like an amoeba with a home. So because they do produce that shell, they're really important carbonate producers, and they produce a large proportion of sediment, and they play a major role in carbon cycling. And although they do perform these great tasks in producing sediment and cycling carbon, they've been largely left out of microbiome studies. So a subset of foraminifera are the larger benthic forams, and these are the topic of my research. And these guys are really cool because they host a variety of algal endosymbionts inside of them. And furthermore, different lineages of foraminifera host different kinds, and you can see them here. I'm going to try and use the clicker there. So we have some diatom symbiont giving it that nice brown color. We have a chlorophyte with green algae symbiont giving it the nice green color. And then we have the rhodophyte symbiont giving it the nice red color. So you can see the diversity represented just in this picture alone. And again, that diversity of those algal endosymbionts gives us an opportunity to see if algal symbiont might be one of those factors that structure the microbiome. And so that's what I'm aiming to investigate. And as well, the few studies that have incorporated foraminifera in microbial research have found that they have diverse microbiomes So we know that they are a species that is viable for this type of research. So here are my study species. They're quite pretty. So the first one at the top is Archaeus angulatus, and you can see from the nice green color that it does host a chlorophyte, which is a green algae. And then we have Cyclobiculina compressa, which also hosts a green algae. Then third, Sorites orbiculus here, and that hosts a dinoflagellate, which is a symbiodinium dinoflagellate, is similar to the one seen in corals. And then lastly, we have Amphistogina gibosa, which hosts a diatom, which you can see from the brown color. So how are these species related? You can see the two that host the green algal symbiont are the most closely related, actually belonging to the same subfamily. And then if you go back a step, you find that they're also related to Sorites orbiculus, and they all belong to the same family of Soritidae. Amphistogina gibosa, my fourth study species, is the most distantly related to the other three. So these guys are pretty similar related, but Amphistogina gibosa has long been considered to be in a different order rotulid order versus the miliolid order, but recent molecular studies have actually identified that it belongs to an entirely different class. So these guys are very distantly related, while these three up here are quite closely related. So the objectives of my thesis research was first and foremost just to see what's there, because there has been no research on Western Atlantic foraminiferal species, and so any investigation into their microbial species will help us understand and compare with Indo-Pacific studies that have looked at the microbial associates of foraminifera. And using that data, I aim to answer three questions about their microbiome structure. So the first hypothesis I had was that more closely related host species would have a more similar microbiome. So the first hypothesis has to do with host species and phylogeny. The second hypothesis is that algal symbiont, and I hypothesized that species with more similar algal symbionts would show a higher similarity in their microbiome. And lastly, I wanted to look at the influence of bleaching on foraminifera, as one of our species does bleach, and so we wanted to see if bleaching would affect the composition of its microbiome, and I hypothesized that the bleached Amphistogina would, of course, have a different composition of microbes than those that are normal or healthy appearing. So why did we make these hypotheses? A lot of our rationales for these hypotheses come from studies of corals and sponges. As I said before, they have the largest data set for us to look at. And the reason I made the first hypothesis is because species is recognized as the strongest driver of microbiome structure in corals and sponges both, and that even accounts for differences in geographic location, in environmental conditions, and seasonal sampling components. And as well, in corals, they found that more closely related coral species tend to have a more similar microbiome than those that are more distantly related. So algal symbiont. So in a study of 11 different types of marine invertebrates and protists, including foraminifera, they studied 2 different types of invertebrates, ones with algal symbionts and those without. And through those 11 different organisms, they found that there was a significant difference between those that had an algal symbiont and those that lacked an algal symbiont, leading researchers to believe that, that algal symbiont is actually providing a unique assemblage of microbes to that organism, and the algae itself might actually have a relationship with microorganisms that it's influencing the microbiome of the host. And like I said before, we have a really great opportunity with foraminifera to address this issue because they host such a wide variety of algal symbionts. So our study incorporates green algae, dinoflagellates, and diatoms, and furthermore, they also host chrysophytes and rhodophytes, which is red algae, but those are not incorporated in this study. And bleaching. When you think of bleaching, you might think of corals, but you can also see it happens in foraminifera. So here we have a normally appearing guy and a partly bleached guy, and then a mostly bleached guy. And you can see the loss of coloration here is due to the loss of the algal symbiont. And coral bleaching studies that look at microbiome of bleached and normal appearing corals find that there is a very strong and tight correlation between microbiome disruption and changes in the microbial associates that happen when the coral bleaches. Amphistogina gibosa, which is this studied species here, has been recognized by our own Reef Indicators Lab to bleach for the last 30 years. But it's important to note that while bleaching in corals is mainly influenced by temperature, bleaching in foraminifera is mainly influenced by photooxidative stress, so that light intensity. So now we're going to talk about my methods. So we have some field methods. We had to extract the DNA, we had to have the DNA sequence, and then we had to analyze that. So let's get to it. So here are field sites. Our first field site was at Tennessee Reef, which, as you can see, is on the Atlantic side of the Florida Reef Tract, and that is where we collected Cyclobiculina compressa and Amphistogina gibosa. The second sampling site on the Florida Bay side of the Florida Reef Tract, and that is the Keys Marine Lab. It's a shallow inlet with seagrass and sand substrate, whereas the Tennessee Reef substrate was coral rubble. So it's also important for you guys to understand that although this one's on the Florida Bay side and this one's on the Atlantic side, there is tidal outflow from the Florida Bay to the Atlantic side, so those water masses are mixing. So the difference is, it's just important to understand that they're not completely separated from each other. They do have the influence of that tidal outflow. And I forgot to mention that we collected Archaeus angulatus and Sorites orbiculus from the Keys Marine Lab site. So how did we get these guys? So divers carried unopened, new, completely brand new Ziploc baggies down to depth where the substrate was going to be collected, and using the highly technical dog poop bag method that anyone with a pet is going to be familiar with, they flipped the bag inside out and placed their hand in it, substrate, flipped it back, and sealed it closed. So then we took that on board the vessel and covered it in seawater and put it in a sealed container and took it straight back to the lab where we could isolate the foram specimens from the substrate. So we took the substrate out of the bag into a stereo microscope where we were able to pick the forams out with sterilized forceps. We then saved the substrate for later, comes back, and we used sterilized using ethanol and flame sterilized brushes. We brushed off the foraminifera to try and get off any surface contaminants and picked it into 0.2 micron filtered seawater. So then once it's in a sterile dish, we repeat the process again for a total of 3 times, each time using a new sterile dish, each time using sterilized forceps, sterilized brushes, and each time using new filtered seawater. So we did this to try and minimize contaminants and just get the foram alone. So in the end, we were left with each foraminifera got to be in its own little vial, completely cleaned of any surface debris. And we also took a substrate sample for each, so that we maintained the pieces that we took off of the foraminifera, and we put that in a separate vial comparison of the substrate microbes versus the microbes associated with foraminifera. And then we flash froze them into liquid nitrogen, where they were transported back here to be stored in a negative 80-degree freezer. So after we got them back to campus, I performed DNA extraction using a handy-dandy DNA extraction kit. We used Qiagen DNeasy, which is a funny name. And when we did the extraction, I used primers that would target that 16S rRNA gene, which is specific to prokaryotes, so that we could target exactly what we wanted to look at. DNA amplification and sequencing was performed by the University of Minnesota Genomics Center, and they used Illumina MiSeq, which is a next-generation sequencing technology using amplicon sequencing and paired end reads. This sequencing targets the V4 region of the 16S gene, which is highly conserved among bacteria and archaea, and it's 250 base pairs long. So from the sequencing facility, what we got back was a microbial library, which is basically the collection of the different sequences that they found inside of each sample. And we use a bioinformatics program to interpret these operational taxonomic units, or OTUs. And an OTU is a way that we group together sequences that are very similar, and then we assign them a taxonomic identity. Data analysis was performed with CHIME. It looks like KIME, but it's CHIME, wherein we picked those OTUs. We assessed taxonomy, calculated diversity metrics, and computed core microbiomes. And what is a core microbiome, you say? Well, you're going to get the answer to that in just a little bit. So we used the statistical software Permanova Plus, which is an add-on to Primer E, which is a non-parametric multivariate statistical test designed for the analysis of ecological data. So that we use that to calculate the dissimilarity between the communities hosted in each host species. So this is what we got back from the sequencing facility. I'm going to go through the different host species and samples just to see what was inside of each of one. So remember those substrate samples that we took for comparison? Those were the largest average sequence reads. So that is the abundance of sequences found in the samples. And they also had the largest number of OTUs. And remember, those are categorizations of similar sequences, and then we assign them an identity. Next up, we had Sorites with the second largest average sequence reads and OTUs. Then Cyclobiculina with around 300,000 average sequence reads and much lower OTUs than Sorites and the environmental samples. Then Archaeus with around 200,000 sequence reads and similar OTUs. And lastly, Amphistogina, both the partially bleached appearing and the normal appearing specimens had the lowest sequence reads, and they also had the lowest amount of OTUs, as you can see here. It's important to note that the normal appearing specimens actually had lower numbers than the partly bleached specimens. So after rarefying the data to make sure that we aren't just seeing differences just because of the large differences in the numbers of sequences, we performed diversity analysis. So this is a PCOA plot, which is a, I just totally blanked on what PCOA stands for. Okay. I had it like 10 seconds ago, but that's okay. So this is a PCOA plot which is used to visualize dissimilarity between samples. And this graph is a weighted unifrac matrix which takes into account abundance and also phylogenetic relatedness of samples and helps to visualize community structure, and in this case, differences in community structure. And the axes represent the percent of variation explained within the data. And you can see that our host species tend to cluster together and are very different from each other. So here we have our Archaeus angulatus in that closed green circle. We have Cyclobiculina compressa in the open green circle. Over down here, very far separated, we have Sorites orbiculus. Primary coordinate analysis. There you go, PCOA. Okay, just came to me. So we have Sorites over down here, and then we have our Amphistogina samples. The open crosses are our partly bleached specimens, and the closed ones are our normal appearing specimens. So each plot point represents a foraminiferal sample, and you can see here they're pretty strongly clustering by host species. So we wanted to test that further, so we performed the Permanova analysis, and we did find that all of our hosts were significantly different. So the analysis did back up our observations. We also performed pairwise tests looking at each, lining them up pair by pair and seeing if they were significantly different, and they all were indeed significantly different, both pairwise and as a whole. So that tells us that yes, our hosts are significantly different, and our data analysis backs that up. So now we're going to get into some more results. When we talk about the composition of a microbiome, we can talk about the whole microbiome, which is every single thing that's in there, and we can talk about the core microbiome, which means that it has to be shared by every single replicate of the group. So when I say an Archaeus angulatus core microbiome, I'm saying that core taxa has to be found in every single Archaeus sample, otherwise it doesn't get to be called core. So we're going to first talk about the whole microbiome. All right, don't get scared. This is a scary plot. We lovingly refer to this as the clown barf chart. What I mainly wanted to show you guys with this image is that our foraminiferal samples in the community structure look different than our substrate samples, which are over here. So as you can see, they're not just mirroring what's in the substrate. They're actually having a different set of community microbes than what is in the substrate. So there you go. And now we are going to look at the whole microbiome by species. So these colors represent bacterial families, and the colors will remain consistent throughout the next slides, so that's something to note. Okay, so first we have Sorites orbiculus, and mainly what is the takeaway from these graphs is that you can see that the replicates of each host species do look similar in terms of what's there, and also they do have similar abundances. So here we see they're looking pretty similar there. Then we have Archaeus, which has a different set of community members, but they do remain consistent throughout the Archaeus samples. And again, Cyclobiculina compressa, the samples look similar. There's a little difference in abundance, but overall, we see a trend there. And then we get to our Amphistogina, and everything kind of just falls apart. See here, we have very little in common here. There are some colors that are the same, and then we have things that just look completely different from each other. So it's important to note that the Amphistogina are much less structured between replicates than are the other species. So now we're going to talk about core microbiomes. So we found 4 OTUs, or 4 microbial taxa, in every single foraminiferal specimen. And we'll talk a little bit about more of them later, but it was interesting to note that we found these 4 in every single sample. And we also found that the numbers of the core taxa for each foraminiferal species was very different. In Sorites, the Sorites samples shared 700 in common, whereas Archaeus angulatus had close to 200, Cyclobiculina had only about 60, Amphistogina, the normal appearing ones, had 18, the partly bleached had 32, and then when you look at the combination of normal appearing and partly bleached specimens, they only share 6 in common. So this graph here, which I'm going to blow up, shows the different bacterial phyla that are represented in the core microbiome of each species. And you can see they're very largely dominated by the phylum Proteobacteria. And there are some very easy to spot similarities between the different foraminiferal species. And we also see that in our Amphistogina gibbosa samples, there's a much larger representation of the bacterial phylum Firmicutes. So I'm just going to go over some key players in each of the core microbiomes. You'll see some of the same key players showing up in the core microbiome of each species. And so I've included 3 or 4 of the top contributing factors to each core microbiome. So for Sorites orbiculus, remember they had 700 core OTUs. We have the family Amoebophiliaceae, family Rhodobacteriaceae, family Flavobacteriaceae, and family Pyrrhululaceae. And it's important to note that this one had 204 OTUs, but only about 10% average abundance, whereas this guy had 31 OTUs, but made up almost half of the abundance of microbes. So then we go to Archaeus. We have 177 OTUs, family Rhodobacteriaceae, family Flavobacteriaceae, genus Rosibium, and family Pyralulaceae. And you might notice that some of them are to the family level and some of them to the genus level. That's because not every single OTU can be identified to the genus level. If we're lucky, they can, but a lot of them only get to family, some of them only get to class. And so we'll get to Cyclobiculina. We see that Rhodobacteriaceae family again. We see that Flavobacteriaceae family again. And this is Pissaricatiaceae. Had to practice that one. All right. So, and that one only had 60 OTUs. So what we did here was to look at species that are more related. Remember, these guys are in the same subfamily. So these are the closest related species that we have in our study. So we looked at the taxa, the microbial taxa that they shared, and we actually found that they shared 31 OTUs. So major constituents in their core microbiome were, again, Rhodobacteriaceae, Flavobacteriaceae, and then this genus Ralstonia. And then we took one step further out and looked at the family of Soritidae, which includes that Sorites orbiculus. So these guys are a little less related than the last slide, but they're still in the same family. And they shared 26 OTUs in common. And again, we see Rhodobacteriaceae, Flavobacteriaceae, Pissaricatiaceae, and genus Rosibium. So we get to the amphistogenic gibosa. These are our normal appearing specimens. And they had 18 OTUs only. And the phylum Firmicutes, which remember was represented more heavily in the amphistogena samples, and with genus Lactobacillus, family Rhodobacteriaceae, genus Ralstonia. Then we go to the partly bleached specimens. They had 32 OTUs with genus Propionibacterium, family Rhodobacteriaceae, phylum Firmicutes, but this time it's genus Streptococcus, and family Pyralulaceae. And then when we get to the combination of bleached and normal appearing, only 6 OTUs, so that's the lowest number. And there's that family Rhodobacteriaceae, genus Propionibacterium, and genus Ralstonia. So why do we care? What is the big deal about the fact that the specimens are sharing a set of core taxa? Well, in microbiome studies, the preservation of microbial taxa across different replicates means that it's probably there for a reason. So we use the fact that it's persisting in the microbiome of multiple species to look for clues as to the possible functions of these microbial taxa. So these are the 4 that we found in every single foraminiferal sample. And that kind of hints at the fact that, hey, there might be a foraminifera in general core microbiome. So are there core taxa? These are the 4 that we found. So Propionibacterium. This is a good candidate for a core bacteria because it's been commonly found in coral core microbiomes. In fact, Dr. Kellogg did a study where they found it, and we actually compared the sequences, and they had a 99% identity match over 239 base pairs. And remember, we targeted 250 base pairs, so that's a pretty good match. So this Propionibacterium has also been observed to occur inside of coral symbiotic dinoflagellates. So that's leading the researchers to hypothesize that this bacterium might actually have a relationship with symbiotic algae. And indeed, we tested a kit blank, I forgot to mention. So we used this to ensure that the persistence of these bacterial taxa were not because of contamination. So what we did was we processed one sample from the DNA sequencing kit without introducing any genetic material. So if everything goes according to plan, there should be nothing in that kit blank sample. So we did, in fact, find that the Propionibacterium that was found in all the foraminifera samples was indeed absent from our kit blank indicating that it's probably a true member of the foraminifera core microbiome. We also found that there was much higher sequencing numbers in the hundreds and thousands of sequences in the foraminiferal samples, while in the substrate samples, Propionibacterium was present, but only at about a few dozen reads. So that's indicating that it's being selectively maintained at higher numbers inside of the foraminiferal specimens. It's not just matching what's in the environment. So Ralstonia, we had 2 OTUs of this. And Ralstonia is also another one of a bacterium that has been found in coral core microbiomes, and it's also another bacterium that has been seen to associate with symbiotic algae. So it seems like a good guess for what it could be in our foraminiferal microbiome. But then when we looked at our kit blank, we found a large number of Ralstonia sequences. So that indicates that this might be due to contamination rather than being a true member. And again, we found much higher numbers in the foraminifera versus the substrate, but that doesn't mean a whole lot because we observed so many sequences and it's a possible contaminant. So Rhodobacteriaceae, the last one. This bacterium has actually been observed to cause disease in algae. So that was a little worrisome when we were looking at the possible functions of this guy. The kit blank contained 6 sequences. However, our foraminifera contained hundreds to thousands. So although it was contained in our kit blank, the abundance was much, much less than the Ralstonia, but it's still worrisome. And we also found that the numbers of sequences between the substrate and between the foraminifera were relatively similar. So this is suggesting a possible environmental influence rather than something being selectively maintained by the host. So when it comes to a foraminiferal core microbiome, Propionibacterium, probably a good candidate, whereas these guys are a little bit more problematic. So in discussing our first hypothesis, we found that more core taxa were shared by more closely related species, with the same subfamily sharing 31 core taxa, the same family, Sorites, sharing 26 core taxa. And that's especially significant because these guys are sharing 84% of their core microbiome in common, even though Sorites has a dinoflagellate symbiont, and these guys both have a green algae symbiont. And then you move back out to different orders. They're only sharing 4, but then again, are they really sharing 4? Remember that 3 of those were a bit problematic. And so this is really suggesting that there is a really heavy influence of phylogenetic relatedness on the structure of the foraminiferal microbiome. We also found that Amphistogina gibosa had a much larger representation of that phylum Firmicutes, and that Amphistogina is also the furthest related member of the foraminifera in our study. It's as we discussed previously, it's in a distinct class from the other 3. And again, here we go with the Firmicutes here. You can see that it's a much larger abundance. And that's actually consistent with findings of one of the few studies that have incorporated foraminifera and microbiome studies. In a study of Amphistogina lobifera, which is in the same genus as Amphistogina, they found that Firmicutes was the overwhelming majority of the core microbiome of their study species. However, it's important to note that Amphistogina lobifera also hosts a diatom symbiont, as does Amphistogina gibosa. So we can't really separate, is it the relatedness, is it the algal symbiont? We need further research to tell us and answer that question. So algal symbiont. We did find that the 2 members of the study that shared the most core microbiome taxa were also host the same type of symbiotic algae, but then again, they're also in the same subfamily. So is phylogenetic relatedness? Is it symbiont? We don't really know. We weren't able to tell for sure in this study. And lastly, bleaching. We did observe some similar patterns to those observed in coral microbiome studies in terms of bacterium that were lost from the host when it bleached. So we found that Staphylococcus bacterium was present in our healthy or normal appearing Amphistogina gibbosa, but was absent from bleached Amphistogina gibbosa. We also found that cyanobacteria was present in our normal appearing samples, while it was not present in our partly bleached samples. And these two are consistent with coral studies where Staphylococcus is observed in healthy corals but not diseased, and also when corals bleach, cyanobacteria is lost or greatly reduced from their microbiomes. However, it's important to note that our healthy appearing individuals only had 0.3% average abundance, so the loss of that amount of diversity is not something that's hugely indicative. And again, although we did have some really nice patterns that were consistent with coral microbiome studies, it's important to note how not much of a pattern there is between our samples. They're just completely all over the place. And we think we might know why. So as I mentioned before, Amphistogina gibbosa, or bleach, and foraminifera in general can bleach, and they do so when they undergo photooxidative stress from light intensity. The onset of the peak bleaching occurs with the peak light incidence, which is around June. We did our sampling in May when it was approaching peak light intensity, and we also had some temperature anomalies the day of our sample collection, and it's actually found that higher temperatures increase the susceptibility of foraminifera to photooxidative stress. So the combination of that higher than normal temperature and the approaching solar maximum could indicate that all of our study specimens of Amphistogina were already thermally stressed. And this is consistent with coral microbiome studies because they actually find that the shift in the microbial composition of the coral occurs before the visual effects of bleaching. So you see in normal appearing corals before they bleach, you see that shift in microbiome. So that crazy diversity might be because we don't really have a healthy individual to compare against, and these weird, crazy lack of a pattern is showing us that they've all been altered. They've all experienced that stress. And we also had some other observations about our samples, and specifically having to do with the way in which our study species make their shells. So there's two types of shell types in this study. There's hyaline, which is this glassy, this shiny, this smooth shell type, which is the Amphistogina gibbosa. And then the other three in the study are porcelaneous. So it's more porcelain-like, it's more opaque, it's not so shiny, it's not so smooth. And as well, the way that they make these shells is different. And the way that these hyaline, these shiny guys, make their shell is they actually cover over themselves, and every time that they make a new chamber, they actually get like a shiny brand new overcoat. And this may provide them with a way to clean their shell from microbes or detritus in the environment, whereas these guys, they don't clean themselves. If a chamber breaks or becomes like infested with something, there's no way for them to really do anything about it, whereas these guys are found to be very, very clean, and every time that they build a new chamber, it's a mechanism by which they can clean off their shell. Furthermore, these guys, the Sorites, they actually produce a polysaccharide glue to adhere themselves to pieces of algae in the environment. And that polysaccharide glue could also provide a place in which microbes could flourish and may contribute to the fact that we saw the highest numbers in these guys, because they are the ones that are producing that glue. So again, bleaching influences much the Amphistogina, but it also affects their shell. So this is a healthy appearing individual. It's that shiny, it's that glassy appearing shell, and it's very smooth. This is a scanning electron microscope image, and you can see that it looks like a polished doorknob. Over here, we have a partly bleached specimen, and their shells often become infested with microbes and become pitted and textured after they have bleaching. And it's important to note that this pitting, this destruction of the shell, only happens after they exhibit that color loss. So this may account for the fact that we saw the higher sequence reads in our bleached appearing ones rather than our healthy ones. So when we go look at the sequence reads in the OTUs, we find that in the foraminiferal samples, the one that produces that polysaccharide glue is having a larger abundance and a larger diversity. And then our shiny, smooth, self-cleaning, normal-appearing guys had the lowest number of sequence reads and OTUs. And then our bleached guys that may have some kind of damage to their shell and thus possible, they're less clean, they have more spaces for microbes to hang on to their shell, we have a little increase in this number of sequence reads. So our conclusions. We're pretty strong on hypothesis 1. We did see some really good evidence that the phylogenetic relatedness is driving microbiomes similarity in more closely related species. For hypothesis 2, we weren't really able to parse out the differences between phylogenetic relatedness and algal symbiont type because more closely related members also shared the same symbiont type. And for hypothesis 3 of bleaching, because we kind of concluded that possibly all of our specimens had been already stressed and we didn't actually have a true healthy comparison, that will definitely require further research. So where could further research go? One interesting factor that could be added onto the study was to include a family that is ancestral to Soritidae, which is family Pneropodidae. And these guys actually host a rhodophyte symbiont, giving them that nice color. And it would continue the kind of decreasing phylogenetic relatedness, and it would be interesting to see if that follows the same patterns that we've observed in these two. As well, larger benthic foraminifera host a variety of algal symbionts, as we've harped on quite a few times. So inclusion of more species that host similar endosymbiont types would provide more clues into how this algal symbiont is structuring the microbiome. So study species such as Leviplaneropoulos, which hosts a chlorophyte similar to Archaeus and Cyclobiculina. There's several species of Amphistogina. One we talked about was Amphistogina lobifera. There's also a few other types, so inclusion of that would be an interesting comparison, as well as Heterostogina depressa, which hosts a diatom symbiont similar to Amphistogina gibosa. As well, any additional research on bleaching should include a seasonal sampling component so they can actually get those true healthy individuals and those bleached individuals. So sampling in January or February would give you a likelihood of getting a truly healthy individual because it's not experiencing that photooxidative stress. I'd like to acknowledge the Cushman Foundation for partial funding through the Student Research Award, the USF College of Marine Science for the Von Rosenstiel Fellowship, as well as Oceanography Camp for Girls Fellowship. I would especially like to thank Dr. Kellogg because she helped me so much with the DNA work, extraction, and use of the laboratory facilities at USGS. Thanks all my committee members, the staff of the Keys Marine Lab, as well as Ben Ross, Sean Beckwith, and Christian Gufader, who were very nice in helping me with my field work. And of course, my family and my friends who have endured a lot of complaining on my part and have been very supportive. And I also like to thank my dog because she's great. We had a permit to do stuff that we did, and you can read more about our stuff online. And thank you guys so much.
Apologies that the dumb major professor forgot to introduce Kellogg and Dr. Breitbart as committee members, but I think you all could have guessed, given the topic, that they were far more instrumental in pulling this off. To tell you how try to get somebody to do something like this ever since sequencing came along. And so finally, this was kind of a last collaboration with Chris and Maya on this. And I also want to acknowledge the mentorship of Teresa Greeley and Angie Lodge for the science education graduate certificate, but also the encouragement and recommendation for her PhD plans. So we've got, it takes a village. So now we'll open to questions from the audience, and then when the audience is finished asking questions, I think most of you know the rules. You can stay and listen to us grill her, or you may discreetly leave. All right. Go ahead, the questions.
Yeah.
Once the shells are pitted after the photooxidative stress, do they then recover to the smooth state after a subsequent period, or are they permanently physically altered?
So with the rotalids, those the amphistogina, like I said, every time that they form a new chamber, it forms around the shell, so they actually have a mechanism by which they can recover from that shell deformation. But they need to recover from the bleaching first. So they actually can die from it if the light intensity is too much or the bleaching occurs too quickly. But yeah, if they do recover from the bleaching, they can repair their shells.
This is more like a future work question. I'm just curious if you took your samples and changed the pH and redid the did the sequencing after at different levels of pH, do you think you might be able to detect changes in your OTUs?
Yeah. So actually, one of the few studies that did incorporate a foraminifera was an experimental study wherein they adjusted the pH of the seawater as well as the temperature and basically stress them out and see what would happen to their microbiome. And they actually, I believe, if I'm remembering the study correctly, they did find a reduction in the number of microbial taxa found in the microbiome of the stressed ones. And I believe they actually found that the reduction was the largest when the effects of temperature and pH were combined. And they did see, I believe, lesser reductions when it was temperature alone versus pH alone. So that's something that has been addressed, but I think, this is a big jumping off point, and there's some room for further studies. Sean?
Nice job, Makenna.
Thank you.
Regarding the kit blank, if a taxa is in the kit blank, is there still some way to weed out that factor and still make comparisons?
Yeah, so like I said, we kind of looked at a few different factors when we were looking at those core taxa. We looked at how high the abundance was in the foraminifera versus the environmental substrate. So that provides clues. It could be a true member, but you really can't say anything for certain if it is found in that kit blank, because there's always the possibility of contamination, and that ruins any certainty that you might really have in that. That's my thoughts.
Great presentation.
Thank you.
So, speaking of blanks, so when I saw Proteobacterium, I'm like, here we go. But, so it's interesting that Grace found it in her corals too. Do you guys know how different those sequences are from human Proteobacterium? Can you tell in that region? Is there any significant differences?
So when I first found the 4 different bacterium in my samples, the first thing I did was I went on BLAST, which matches the sequences, and when I copied and pasted, I came back with all this human skin bacteria, and I was like, oh crap, I just totally screwed this up. And I think the reason that those comes up is just because there's a larger proportion of those data points. There's more human entries into the database. But I would have to look and actually do like a sequence side-by-side comparison. The only one I did actually compare quantitatively was to the coral microbiome one, but it would be interesting to see how much they do overlap.
Yeah, if you can tell with that.
Yeah.
It's interesting because it's one of the core members of healthy
Mhm.
humans.
Yeah.
So it's interesting that it's healthy coral.
Yeah.
Healthy flora.
Definitely.
And there also seems to be a correlation between the number of sequences and the diversity that you found. So
Mhm.
the more sequences you got, the more diversity you found. So once you normalize for that, do you still see significant differences? Like the beach is still like more diverse than anything else once you normalize your data? I'm blanking on the word. So we rarefied to account for the abundances, and so we rarefied down to the lowest sequencing observed, which was around 20,000, and then we performed the analyses of the diversity metrics, and it takes into account the phylogenetic relatedness as well as the abundance of different community members, and we still did find that the hosts were significantly different in terms of were they more diverse once you rarefied the data. I don't exactly have an answer to that. I don't think that's an analysis that we performed, but it would be something to look at for sure.
Good job. Any other questions? All right, well, good timing? Be sure and have a good one on your way out.
Yeah, take some food. I don't want to take it home.